Hello everyone, my name is Neil Faulkner and today we are going to cover the topic of biological macromolecules and lipids. So just an overview of the session then. Macromolecules are polymers and they're built from monomers. Carbohydrates serve as fuel and building material. Lipids are a diverse group of hydrophobic molecules. Proteins include a diversity of structures resulting in a wide range of functions. Nucleic acids store, transmit and help express hereditary information. And then finally, genomics and proteoeconomics have transformed biological inquiry and applications. So it's said that macromolecules are polymers built from monomers. So if we look at large carbohydrates, proteins and nucleic acids, they're also known as macromolecules for their huge size. The chain-like molecules are called polymers. A polymer is basically a long molecule consisting of many similar or identical building blocks linked by covalent bonds, so much as a train consists of a chain of boxcars. The repeating units that serve as the building blocks of a polymer are smaller molecules called monomers. In addition to forming polymers, some monomers have functions of their own. So first of all, we need to look at the synthesis and breakdown of polymers. Although each class of polymer is made up of a different type of monomer, the chemical mechanisms by which cells make polymers and break them down are similar for all classes of large biological molecules. In cells, these processes are facilitated by enzymes, specialized macromolecules that speed up chemical reactions. The reaction that connects a monomer to another monomer or polymer is called a condensation reaction, a reaction in which two molecules are covalently bonded to each other with the loss of a small molecule. If a water molecule is lost, it is known as a dehydration reaction. For example, carbohydrates and protein polymers are synthesized by dehydration reactions. Each reaction contributes a part of the water molecule that is released during the reaction. One provides a hydroxyl group, while the other provides a hydrogen. The reaction is repeated as many times as possible, and this is added to the chain one by one, gradually lengthening the polymer. Polymers are disassembled to monomers by a mechanism known as hydrolysis. This is a process that is essentially the reverse of the dehydration reaction. Hydrolysis means water breakage. The bond between monomers is broken by the addition of a water molecule with a hydrogen from water attaching to one monomer and the hydroxyl group attaching to the other. An example of hydrolysis within our bodies is the process of digestion. The bulk of organic material in our food is formed by polymers that are much too large to enter our cells. So within our digestive tract, various enzymes attack the polymers, speeding up hydrolysis. Released monomers are then absorbed into the bloodstream for distribution to all body cells. Those cells can then use dehydration reactions to assemble the monomers into new different polymers that can perform specific functions required by the cell. Dehydration reactions and hydrolysis can also be involved in the formation and breakdown of molecules that are not polymers, such as lipids. So the diversity of polymers. A cell has thousands of different macromolecules. The collection varies from one type of cell to another. The inherited difference between close relatives, such as human siblings, reflects small variations in polymers, particularly DNA and proteins. Molecular differences between unrelated individuals are much more extensive, and those between species greater still. The diversity of macromolecules in the living world is vast, and the possible variety is effectively limitless. So then, what is the basis for such diversity in large polymers? Building a huge variety of polymers from such a limited number of monomers is analogous to the construction of hundreds of thousands of words from only 26 letters of the alphabet. The key here is arrangement, the particular linear sequence that the units follow. However, this analogy falls far short of describing the great diversity of macromolecules because most biological polymers have many more monomers than the number of letters in even the longest word. Proteins, for example, they're built from 20 kinds of amino acids arranged in chains that are typically hundreds of amino acids long. The molecular logic of life is simple, but it's very elegant. It's comprised of small molecules common to all organisms, and these act as the building blocks that are then ordered into unique macromolecules. Carbohydrates, they serve as fuel and building material. The simplest carbohydrates are the monosaccharides, or simple sugars. These are the monomers from which more complex carbohydrates are built. Disaccharides are their double sugars. They consist of two monosaccharides joined by a covalent bond. Carbohydrate macromolecules are polymers called polysaccharides composed of many sugar building blocks. So then, if we look at monosaccharides, they're the simple sugars. They generally have their molecular formulae that has some multiple of their unit, so CH20. Glucose, the formula for that is C6H12O6. The most common monosaccharide is of central importance in the chemistry of life. In the structure of glucose, we can see the structure of a monosaccharide. The molecule has a carboxyl group, C=0, and multiple hydroxyl groups, OH. Depending on the location of the carboxyl group, a monosaccharide is either an aldose or a ketose. Another criterion for classifying monosaccharides is the size of the carbon skeleton, which ranges from 3 to 7 carbons long. Glucose, fructose, and other sugars that have 6 carbons are called hexases. Trioses and pentoses are also common. Another source of diversity of sugars is the way in which their parts are arranged, specially arranged symmetric carbons. What seems like small differences is significant enough to give two sugars distinctive shapes and building activities, thus different behaviours. Monosaccharides, particularly glucose, they are major nutrients for our cells and the human body. In the process that we will know and we'll cover later is cell respiration. So basically cells extract energy from glucose molecules by breaking them down into a series of reactions. Well, not only are monosaccharides a major fuel for cellular work, but their carbon skeletons also serve as raw material for the synthesis of other types of small organic molecules such as amino acids and fatty acids. Monosaccharides that are not immediately used are generally incorporated as monomers into disaccharides or polysaccharides. So if we look at disaccharides, then slightly more complex sugars, these consist of two monosaccharides joined by a glycosidic linkage, a covalent bond formed between two monosaccharides by a dehydration reaction. For example, maltose is a disaccharide formed by the linking of two molecules of glucose. Maltose is used as an ingredient in brewing beer. The most prevalent disaccharide is sucrose, or table sugar, its two monomers are glucose and fructose. Plants generally transport carbohydrates from leaves to their roots and other non-photosynthetic organs in the form of sucrose. Lactose, the sugar present in milk, is another disaccharide. In this case, a glucose molecule is joined to a galactose molecule. Disaccharides must be broken down into monosaccharides to be used for energy for organisms. So with humans, there's such a thing as lactose intolerance. And this is a common condition in humans who lack lactase, the enzyme that breaks down lactose. The sugar is instead broken down by intestinal bacteria, causing the formation of gas and subsequent cramping. The problem with lactose intolerance may be avoided by taking the enzyme lactase when eating or drinking dairy products, or consuming dairy products that already have been treated with lactase to break down the lactose. Polysaccharides then, these are macromolecules, polymers with a few hundred to a few thousand monosaccharides joined by glycosidic linkages. Some polysaccharides are hydrated as needed to provide monosaccharides for cells. Other polysaccharides serve as building material for structures that produce a cell or the whole organism. The architecture and function of a polysaccharide are determined by its monosaccharides and by the patterns in the glycosidic linkages. Both plants and animals use these sugars later in the form of storage of polysaccharides. Plants store starch as a polymer of glucose monomers. Synthesizing starch enables the plant to stockpile surplus glucose. Because glucose is a major cellular fuel which represents stored energy, the sugar can later be broken down by this plant from this carbohydrate bank by hydrolysis, which breaks down the bonds between the glucose monomers. Most animals, including humans, also have enzymes that can hydrolyze plant starch, making glucose available as a nutrient for cells. The fruits of wheat, rice, and others are major sources of starch in the human diet. Animals store a polysaccharide called glycogen, a polymer of glucose that is like amylopectin, but now more extensively branched. Vertebrates store glycogen mainly in liver and muscle cells. Breakdown of glycogen in these cells releases glucose when the demand for energy increases. This stored fuel cannot be stored in an animal for long, however, so there are disadvantages to that. In humans, most glycogen stores are depleted in almost a day unless they are replenished by eating. This is an issue of concern in low, in which can result in weakness and fatigue. Structural polysaccharides, these are enzymes that digest starch by hydrolyzing its A linkages, but unable to hydrolyze the B linkages of cellulose due to the different shapes of these two molecules. In fact, few organisms possess enzymes that can digest cellulose. Almost all animals, including humans, cannot digest cellulose. The cellulose in our food passes through the digestive tract and is eliminated with the feces. The cellulose abrades the wall of the digestive tract and stimulates the lining to secrete mucus, which aids in the smooth passage of food through the digestive tract. Thus, although cellulose is not a nutrient for humans, it is an important part of a healthy diet. Most fruits, vegetables, and whole grains are rich in cellulose. On food packages, insoluble fibre refers mainly to cellulose. So some microorganisms can digest cellulose, breaking it down into glucose monomers. A cow harbours cellulose digesting prokaryotes and protists in the gut. These microbes hydrolyze the cellulose of hay and grass and convert the glucose into other compounds that nourish the cow. A termite, which is unable to digest cellulose by itself, has prokaryotes and protists living in its gut that can break a small meal out of wood. Some fungi can also digest cellulose and store it elsewhere, thereby helping recycle chemical elements within Earth's ecosystems. Another important structural polysaccharide is chitin. This is the carbohydrate used by insects, spiders, crustaceans, and other related animals to build their exoskeletons. These are hard cases that surround the soft parts of the animal. Chitin is found in fungi, which uses this polysaccharide rather than cellulose as the building material for their cell walls. Chitin is similar to cellulose with B linkages, except that the glucose monomer or chitin has a nitrogen-containing attachment. Lipids are the one class of large biological molecules that do not include true polymers, and they are generally not big enough to be considered macromolecules. The compounds called lipids are grouped with each other because they share an important trait. They are hydrophobic, which means they mix poorly, if at all, with water. This behavior of lipids is based on their molecular structure. Although they may have some polar bonds associated with oxygen, lipids consist mostly of hydrocarbon regions with relatively non-polar CH bonds. Lipids are varied in form and function. Although fats are not polymers, they are large molecules assembled from smaller molecules by dehydration reactions. A fat consists of a glycerol molecule joined by three fatty acids. Glycerol is an alcohol which each of its three carbons bears a hydroxyl group. A fatty acid has a long carbon skeleton, usually 16 or 18 carbon atoms in length. The carbon at one end of the skeleton is part of a carboxyl group, functional group that gives these molecules their name fatty acids. The rest of the skeleton consists of a hydrocarbon atom, a hydrocarbon chain. The relatively non-polar CH bonds in the hydrocarbon chains of fatty acids are the reasons why fats are hydrophobic. Fats separate from water because the water molecules and hydrogen bond to one another and exclude the fats. This is why vegetable oil separates from the aqueous vinegar solution into a bottle of salad dressing. So if we look at saturated fats and unsaturated fats, terms unsaturated fats and saturated fats are commonly used in the context of nutrition. These terms refer to the structure of hydrocarbon chains of the fatty acids. If there are no double bonds between carbon atoms comparing a chain, then as many hydrogen atoms as possible are headed into the carbon skeletons. Such a structure is said to be saturated with hydrogen, and the fatty acids is said to be used by an unsaturated fatty acid. A fat made from saturated fatty acids is called a saturated fat. Most animal fats are saturated. The hydrocarbon chains of their fatty acids, the tails of their fat molecules, lack double bonds, and their flexibility allows the fat molecules to pack together tightly. Saturated animal fats, such as lard and butter, are solid at room temperature. An unsaturated fatty acid has one or more double bonds with one fewer hydrogen atoms on each double bonded carbon. In contrast, the fats of plants and fishes are generally saturated, meaning that they are composed of one or more types of unsaturated fatty acids, usually liquid at room temperature. Plant and fish fats are referred to as oils. Cod liver oils are examples, and so is olive oil. The phrase hydrogen and vegetable oils on food labels means that the unsaturated fats have been synthetically converted into saturated fats by adding hydrogen, allowing them to solidify. Peanut butter, margarine, and many other products are hydrogenated to prevent them from separating out in liquid form. A diet rich in saturated fats is one of several factors that may contribute to cardiovascular disease known as atherosclerosis. In this condition, deposits called plaques develop within the cells of blood vessels, causing inward bulges that impede the blood flow and therefore the resilience of the blood flow system. So if we look at trans fats, the process of hydrogenating vegetable oils produces not only saturated fats but also unsaturated fats with trans double bonds. It appears that trans fats can contribute to coronary heart disease. Trans fats are commonly found in baked goods and processed foods. In the USA, the Food and Drug Administration requires nutritional labels to include information on trans fat content. In addition, the FDA has ordered US food manufacturers to stop producing trans fats by 2021. The major function of fats is energy storage. The hydrogen chains of fats are similar to gasoline molecules and just as rich in energy. A gram of fat stores twice as much energy as a grain of a polysaccharide such as starch. Because plants are relatively immobile, they can function with bulky energy storage in the form of starch. Vegetable oils are generally obtained from seeds, where more compact storage is an asset to plants. Animals must carry their energy storage with them, so there is an advantage to having a more compact reservoir of fuel fat. Humans and other mammals stock their long-term food reserves in adipose cells, which swell and shrink as fat is deposited and withdrawn from storage. In addition to storing energy, adipose tissue also cushions vital organs such as the kidneys, and a layer of fat beneath the skin insulates the body. So in terms of phospholipids, cells as we know them could not exist without another type of lipid called phospholipids. Phospholipids are essential for cells because they are major constituents of cell membranes. Their structure provides a classic example of how fats function at the molecular level. A phospholipid is similar to a fat molecule, but the only difference is the fatty acids attached to glycerol rather than three. So there's two fatty acids attached to glycerol rather than three. The third hydroxyl group of glycerol is joined to a phosphate group which has a negative electrical charge in the cell. Typically, an additional small charged or polar molecule is also linked to the phosphate group. Choline is one such molecule, but there are many others as well, allowing formation of a variety of phospholipids that differ from each other. So the tails of phospholipids show different behaviours with respect to water. Their hydrocarbon tails are hydrophobic and are powered from water. However, the phosphate group and its attachments form a hydrophilic head that has an affinity for water. At the surface of the cell, phospholipids are arranged in a similar bilayer. The hydrophobic heads of the molecules are on the outside of the bilayer, in contrast with the aqueous solutions inside and outside of the cell. The hydrophobic tails point towards the interior of the bilayer, away from the water. The phospholipid bilayer forms a boundary between the cell and its external environment and enables separate compartments within eukaryotic cells. The existence of cells depend on the properties of phospholipids. So steroids, these are lipids characterized by a carbon skeleton consisting of 4 fused rings. Different steroids are distinguished by a particular chemical group attached to this ensemble of rings. Cholesterol is a type of steroid. It's a crucial molecule in animals. It's a common component of animal cell membrane and is also a precursor from which other steroids such as sex hormones are synthesized. In vertebrates, cholesterol is synthesized in the liver and is also obtained from the diet. A high level of cholesterol in the blood may contribute to atherosclerosis although some researchers are questioning the roles of cholesterol and saturated fats in the development of the condition. So when we're looking at proteins, we look at how they have diversity of structures, and this subsequently results in a wide range of functions. Nearly every dynamic function of a living being depends on proteins. Proteins account for more than 50% of the dry mass of most cells, and they are instrumental in almost everything organisms do. Some proteins speed up chemical reactions, while others may play a role in defense, storage, transport, cellular communications, movement, or structural support. Life would not be possible without enzymes, much of which are proteins. Enzymatic proteins regulate metabolism by acting as catalysts, chemical agents that selectively speed up chemical reactions without being consumed in the reaction. Because an enzyme can perform its function over and over again, these molecules can be thought of as workhorses that keep cells running by carrying out the processes of life. Proteins are all constructed from the same set of 20 amino acids linked by an unbranched polymer. The bond between amino acids is called a peptide bond, so a polymer of amino acids is called a polypeptide. A protein is a biologically functional molecule made up of one or more polypeptides, each folded and coiled into a specific 3D structure. So when we're looking at amino acids represent the monomers. All amino acids share a common structure. An amino acid is an organic molecule with both an amino group and a carboxyl group. The small figure shows the general formula for an amino acid. At the center of the amino acid is an asymmetric carbon atom called the alpha carbon. In 4 different partners are an amino group, a carboxyl group, a hydrogen atom, and a variable symbolized by it. The R group, also called the side chain, differs with each amino acid. The R group may be as simple as a hydrogen atom, or it may be a carbon skeleton with various functional storage attached. The physical and chemical properties of the side chain determine the unique characteristics of a particular amino acid, thus affecting its role in a polypeptide. The amino acids are grouped according to the properties of their side chains. One group consists of amino acids with nonpolar side chains, which are hydrophobic. Another group consists of amino acids with polar skin chains, which are hydrophilic. There are 20 amino acids that cells use to build their thousands of proteins. Acidic amino acids have side chains that we generally negative in charge due to the presence of a carboxyl group, which is usually dissected at cellular pH. Basic amino acids have an amino group in their side chains that are generally positive in charge because they are acidic, and basic side chains are also hydrophilic. So when we look at polypeptides, now that we've examined amino acids, let's see how they are linked to form polymers. When 2 amino acids are positioned so that the carboxyl group of one is adjacent to the amino group of another, they can become joined by a dehydration reaction with the removal of a water molecule. The resulting covalent bond is called a peptide bond. Repeated over and over, this process yields a polypeptide, a polymer of many amino acids linked by peptide bonds. The repeated sequence of atoms is called the polypeptide backbone. Extended from the backbone are the different side chains, R groups of amino acids. Polypeptides range in length from a few amino acids to 1,000 or more. Each specific polypeptide has a unique linear sequence of amino acids. Note that one end of the polypeptide chain has a free amino group, while the opposite end has a free carboxyl group, the C terminus. The chemical nature of the molecule as a whole is determined by the kind of sequence of the side chain, which determine how a polypeptide folds and thus its final shape and chemical characteristics. The immense variety of polypeptides in nature illustrates the important concept introduced earlier, that cells can make many different polymers by linking a limited set of monomers into diverse sequences. So in terms of protein structure and function, the specific activities of proteins results from their intricate 3-dimensional architecture, the simplest level of which is the sequence of their amino acid. A functional protein is not just a polypeptide chain, but one or more polypeptides practically twisted, folded, and coiled into a molecule of a unique shape, which can be shown in several different types of models. It is the amino acid sequence of each polypeptide that determines what 3-dimensional structure the protein will have under normal cellular conditions. When a cell synthesizes a polypeptide, the chain that links spontaneously, assuming the functional structure for that protein. This folding is driven and reinforced by the formation of various bonds between parts of the chain, which in further depends on the sequence of amino acids. Many proteins are roughly spherical, i.e. globular proteins, while others are shaped like long fibers, i.e. Even with these two categories, countless variations exist. In almost every case, the function of a protein depends on its ability to recognize and build other molecules. For example, the exact shape between and morphine, heroin, and other opiatic drugs are able to mimic endorphins because they all have a shape similar to that of endorphins, and these endorphins can sort of like fit into the parts of the brain that boost your mood. So it said there are 4 levels of protein structure which you need to know about. In spite of their great diversity, proteins share this superimposed level of structure, known as primary, secondary, and tertiary structure. A 4th level, quaternary structure, arises when a protein consists of 2 or more polypeptide chains. When we look at the primary structure says that a protein is a sequence of amino acids. As an example, let's consider transthyretin. As this is a globular blood protein that transports vitamin A and one of the thyroid hormones. Transthyretin is made up of 4 identical polypeptide chains, each composed of 127 amino acids. Each of the 127 positions along the chain is occupied by one of the 20 amino acids indicated by its 3-letter abbreviation. The primary structure is like the order of letters in a very long word. If left to chance, there would be 20 to the power of 127 different ways of making a polypeptide chain, 127 amino acids long. However, the precise primary structure of a protein is determined not by the random linking of amino acids, but by inherited genetic information. The primary structure in turn dictates its secondary structure and tertiary structure due to the chemical nature of the backbone and the side chains of the amino acids along the polypeptide. So when we look at the secondary structure, most proteins have segments of their polypeptide chains repeatedly coiled or foiled in patterns that contribute to the protein's overall shape. These coils and folds, collectively referred to as the secondary structure, they're the result of hydrogen bonds between the repeating constituents of the polypeptide backbone. Within the backbone, the oxygen atoms have a partial negative charge, and the hydrogen atoms attached to the nitrogens have a partial positive charge. Therefore, hydrogen bonds can form between these atoms. One such secondary structure is the helix, a delicate coil held together by hydrogen bonding between every 4th amino acid. The other secondary structure is the B-pleated sheet. In terms of tertiary structure, these are superimposed on the patterns of secondary structure and the protein's tertiary structure. The tertiary structure is the oval shape of a polypeptide resulting from interactions between the side chain, the R groups of the amino acids. One type of interaction that contributes to a tertiary structure is called a hydrophobic interaction. As a polypeptide folds into its functional shape, amino acids with hydrophobic side chains usually end up in clusters at the core of the protein out of contact with water. Thus, the hydrophobic interaction is actually caused by the exclusion of non-polar substances by water molecules. One non-polar amino acid side chains are close together. Van der Waals interactions can help hold them together. Meanwhile, hydrogen bonds between polar side chains and ionic bonds between positively and negatively charged side chains also stabilize the tertiary structure. And finally, the quaternary structure. Some proteins consist of 2 or more polypeptide chains aggregated into one functional macromolecule. Quaternary structure is the overall protein structure that results from the aggregation of these polypeptide subunits. Hemoglobin, the oxygen-binding protein of red blood cells, is another example of a globular protein with quaternary structure. It consists of 4 polypeptide subunits, 2 of one kind A and 2 of another kind B. Both A and B subunits consist primarily of a helical secondary structure. Each subunit has a non-polypeptide component called heme with an iron atom that binds oxygen. So finally, when we look at humans again, there's a disease known as sickle cell disease. Even a slight change in the primary structure can affect a protein's shape and the ability to function. For instance, sickle cell disease, which is an inherited blood disorder, is caused by the substitution of one amino acid for the normal one at the position of the 6th amino acid in the primary structure of hemoglobin, the protein that carries oxygen in red blood cells. The normal red blood cells are disc-shaped, but in sickle cell disease, the abnormal hemoglobin molecules tend to aggregate into chains, deforming some of the cells into sickle shape. A person with this disease has periodic sickle cell crises when the angular cells clog tiny blood vessels, impeding blood flow. The toll taken on such patients is a dramatic example of how a simple change in protein structure can have devastating effects on protein function. So what then determines protein structure? The folding normally occurs as the protein is being synthesized in the crowded environment within the cell, aided by other proteins. However, protein structure also depends on the physical and chemical conditions of the protein environment. If the pH, salt concentration, temperature, or other aspects of its environment are altered, the weak chemical bonds and interactions within the protein may be destroyed, causing the protein to unravel and lose its native shape, a change called denaturation. Because it is misshapen, the denatured protein is biologically inactive. Most proteins become denatured if they are transferred from an aqueous environment to a non-polar solvent. The polypeptide chain refolds so that its hydrophobic regions face outward toward the solvent. Denaturation can also result from excessive heat, which agitates the polypeptide chain enough to overpower the weak interactions that stabilize the structure. The white of an egg becomes opaque during cooking because the denatured proteins are insoluble and solidify. This also explains why excessively high fevers can be fatal. Proteins in the blood tend to denature at very high temperatures.